So, good morning and welcome to the 2014 IMI Stakeholders Forum. It's really a pleasure to see you all in the room. And I would also like to welcome those following us via web streaming. So this meeting is indeed a landmark in the lifetime of IMI, as this is the last Stakeholders Forum organized under the first phase of the initiative. And you know that it's a unique public-private partnership between the European Commission and the European Federation of Pharmaceutical Industries and Association. And based on the achievement of the first phase, fortunately, a second phase is about to be launched in coming weeks. And what is quite remarkable, it will be with an increased budget over 3 billion euros. So we decided to dedicate the whole afternoon session to this exciting new initiative, which, as you know, is named IMI2. So this morning, we will focus on IMI1, which, as you also know, is very much dedicated to the development of new methods and tools to improve the assessment of drug efficacy and drug safety. So you will find in your meeting bag a brochure which presents the main achievements of IMI1 so far. And as you will realize, induced pluripotent stem cells, IPS, are at the core of several IMI projects. Actually, we directly dedicate around 100 million euros to two major projects directly related to IPS, but we have also a number of other projects which make use of IPS, as we will hear during this morning session. Indeed, IPS emerged during the last few years as one of, if not the major cross-cutting themes in the development of new strategies for therapeutic innovation. And again, this will be the subject of our presentation and discussion this morning. So to set the scene for this IPS presentation, it's my pleasure to introduce our keynote speaker, Ole Isacson. So Ole is professor of neurology at Harvard Medical School and a founding director of the Neuroregeneration Research Institute at the McLean Hospital. His work is focused on the understanding and treatments of neurodegenerative diseases, and he's also principal faculty at the Harvard Stem Cell Institute. So he's really in an ideal position to educate all of us about IPS and all their applications in drug innovation. Ole, I think that I can say that you are a true IMI friend. You assist us in the evaluation of several IMI projects, and I think that your presence this morning is another evidence of this friendship. So thank you very much for being with us, and without further ado, I will give you the floor to deliver your lecture on the use of stem cells in drug development.
Thank you, Michel. It's indeed a great honor to be here. I would like to thank the organizers, of course, for making me an invitee, but also to spell out to you that from an international perspective, the IMI is really, as Professor Goldman has mentioned many times, an experiment. And it's viewed all over the world with high hopes to be an example of public-private partnerships. So nobody else has really tried this at this level before. And I really commend all of you to have taken this on. So my presentation today is to give you a global perspective on stem cells. As we just heard, IPS cells, induced pluripotent stem cells, are now an opportunity both in science and medicine and drug discovery to use a new tool. And IMI really has become really a world leader in initiating some of these new programs. So first, I'd like to tell you a little bit about stem cells. In general, The world is facing a different kind of problem in medicine than in decades before, primarily because many populations are aging healthily, we hope, but in some cases, of course, aging is obviously the process that leads to the diseases that you see here. Aging processes affect many systems: heart and vascular disease, diabetes, my specialty, Parkinson's disease, and many neurological diseases. And as such, represent an enormous burden for societies worldwide and something that we must address before this avalanche. So what about stem cells? My kids and my students always ask me why I work on regeneration and simply not just replace entire body parts. Actually, this is possible. In this flatworm here you see on a wall of an aquarium, if you cut off the head or the tail, in a few days after you've made that cut, at the top of the cut you see stem cells. And these stem cells are very immature stem cells, and they actually grew a new head or a new tail within about 10 days or so. And this is illustrated, as you can see on this side here. We wish the human body functioned that way, but that's actually not at all the case. And part of the theme here about human iPS cells is exactly that. There are differences between cells and animals and cell types. And this is another very interesting example. That's one of my favorite articles, actually from the New York Times. It's based on an article from a colleague of mine, former colleague or friend, Jonas Frisén at Karolinska Institute in Stockholm. What does it show? It says, an eye is forever, but is a liver. And we all have stem cells in our bodies. In particular, for example, as we hear here, in the liver. The liver is replaced at a certain rate every year. And also, for example, muscle. In your muscle, there are a replacement of cells completely in about 15 years. And the entire bone of a person is actually replaced every 10 years or so. But the eye and the brain does not replace its cells, which connects to a concept we call as regenerative medicine. There are certain body parts and certain cell systems that don't regenerate, unlike the flatworm. So, in a global perspective, stem cells are important for understanding the way the body deals with disease, but also growth. And those are the things that are very active in science today, and there are hundreds of studies underway at this minute elsewhere in the world and here to address those. But what IMI did smartly was to understand the opportunity from the FPI perspective as well of studying disease and drug screening from the perspective of iPS cells. And in that way, we can study mechanisms of disease, drug screening, and to some extent cell therapy, which is not the topic here. So what is a stem cell and an iPS cells? First, I'm going to show you what we don't work on. The embryonic stem cell, though, is the model for an iPS cell. And an embryonic stem cell is defined as an embryo that's fertilized. And inside that, in a few days, you get something called a blastocyst. And that blastocyst has some cells inside, which you can grow in a dish. And essentially, those cells are totipotent, or pluripotent, or basically can become any cell in the body. Therefore, if we develop culture conditions, we can make these cells into any cell we desire. Now, this happens to be very difficult because we have to simulate what normally happens in a body, and that requires a lot of research to accomplish. So what about human-induced pluripotent stem cells? What are they? In Japan, not long ago, in about 2006, 2007, a team led by Dr. Yamanaka in Kyoto discovered while they were working on cancer that if they started to expose cells in the body, those are adult cells like your skin or blood, those are what we call fully differentiated, they are not stem cells. If you expose them to 3 or 4 factors, you can make them into that embryonic stem cell I just showed you. And that led to him, Yamanaka, that is, being awarded the Nobel Prize a few years ago. And nowadays, we can do this reprogramming, that is called, by using various molecular mechanisms. But they all have the same idea of putting in these 2 to 3 factors in the cell over about 4 or 5 days, sometimes more. And eventually, the cell will lose all its programming and become naked and behave just like an embryonic stem cell. And then that's the problem. So why are these stem cells so important to us? Well, there are many reasons, but let's look at one in particular. Most research in the world up to this point have been done on animal cells, whereas obviously drugs are delivered to humans. And that's at the heart of this problem. And heart, no pun intended, is what we're looking at here. Your heart beats, every second or so, and it has an electrocardium structure like this. We call this, there's a P, when the arterial node starts like this. Every time you beat, you have this heartbeat, and this time here is called QT. A mouse heartbeat is quite different than a human heart. It is much faster and has a very different plateau. So, for example, in a human, while a heartbeat happens this way, it will be 5 times faster in a mouse. And the ion current that depolarize the heart, it's like a muscle, and they repolarize, are very different in the heart, in the mouse, than in the human. And this has very clear consequences. Indeed, the number of medicines withdrawn from the market due to cardiac toxicity is very high. Here, from a paper by Brahms et al., you see that there are 11 or so, and these were very significant drugs on the market. Some of them actually, recently, had very severe side effects, like Vioxx, the anti-inflammatory drug, and caused myocardial infarction. Many of these drugs, in my opinion, could have been prevented from entering the market if they had started with human iPS cells. And that's just the tip of the iceberg. There's another 20 or 25 drugs or so that have a black label. That means that when you take the drug, it says you better be aware of this, but this drug can hurt you. And it's usually with arrhythmias. And arrhythmias, as you know, if your heart flutter enough time, it will not pump enough blood to you, and you may actually die. So this is a very serious concern, and it can partly be addressed by these stem cells. So in other words, what we've talked about so far is this. A patient, or you and I, have various cells that can be reprogrammed into iPS cells that can then be developed into specialized cells, and they can model disease, screen for drug, or even check for toxicity, such as liver, heart, and others. In a sense, if we see an arrhythmia in those cells, which we can see in a dish, we would prevent them from going further, or even, as some people have said in the industry, we could run a clinical trial in a dish or in a tube by testing various individual cells and see what their response is. And hopefully, in that way, as we will hear later this afternoon or later today from Ruth McKernan, drugs can be developed that are more specific and effective. So what are the global advancement of medical research and treatment based on these? And what are the latest development of stem cell research? I'd like to give you a few examples and try to convey to you what I think are the most exciting parts in the fields. This is a slide from a Japanese colleague who used to work in my laboratory, Dr. Haruhisa Inoue, who had a publication not long ago in Science Translational Medicine. What he and his team developed in Kyoto was models of Alzheimer's disease. AD here stands for Alzheimer's disease. He took the cells from patients in a population of Alzheimer's disease, some with a specific mutation that made them likely to develop the disease, some just that had Alzheimer's disease without the genetic familial known link. He reprogrammed them and grew them in a dish into these human neurons. Then he studied them for disease modeling, and that's what the scientists typically do these days. And when he grew them in a dish, he noticed that in these 4 examples, which is just 4 examples, that some of these cells developed abnormalities with this mutation, and at this early stage, about 60 days in a cell culture, these cases of this human here from a naturally developing sporadic form developed. He also found changes in some of the cells in the brain, and he also found this clogging protein called amyloid plaque that is generated by this gene in some of these cells, in these individual cells. The cells showed signs of stress, meaning they were adapting to something maybe pathological. And they also had to respond to a drug. I actually spoke to him why he chose, on earth would you choose this drug? And he says, well, he said, in Japan we use fish oil. And indeed this is a derivative of fish oil, which is maybe many of you take alpha omega-3, and some of those fish oil responses reduced some of these cellular stresses. Just as a scholastic example, not one that we think we need to patent. Another thing that I know more about is Parkinson's disease, and I'd like to illustrate to you how iPS cells have been used there. Now remember, we were working on Parkinson's disease before iPS cells arrived on the scene, but we have used them since, and I'd like to illustrate that. Here is a person's brain, and somewhere around here is the midbrain. And they contain cells like these. This is a slice from my hometown, Boston. And this is actually from a patient without the disease. It looks like a bit like a shape like this. And this black substance here contains about 1 million dopamine neurons in a person's brain. We're born with these. And as we grow older or are affected by disease, here you see them again, there's various population of these so-called dopaminergic neurons that die. And over the years, we've defined this red population as the most vulnerable cell, the most risky cell you have in your brain for developing this disease. We know a lot about this cell, and we know a lot about why it may degenerate, but we have no treatments to prevent that. And to illustrate the problem, for both Parkinson's disease, Alzheimer's disease, ALS, and others, what the healthcare and I should say all medicine is involved in is understanding what we call the preclinical phase. Whether you like to know it or not, many of us are developing heart disease before we know it, Alzheimer's disease before we know it, or Parkinson's disease before we know it. And this preclinical phase, if you start with 1 million dopamine neurons here, goes on for quite a while. With aging, in fact, the problem is so considerable that most people that live beyond the age of 100, when looked by a neuropathologist, and I know one who told me this, they all show signs of these diseases. Whether it's broken through, though, to become symptomatic is another thing. And the key message for you to take home, perhaps about neurology today, is that we always have a reserve capacity in the brain. And that's not just humans, but also animals. So until you lose these number of cells, about 70% of them, you don't show disease. So the worldwide effort in science now is very reasonable and logical. We are trying to find biomarkers to come to this red zone and try to prevent disease before it happens or slow it down. Obviously, we do that in many ways. We will hear about small molecule research that we do, gene therapy and cell therapy. But this is a very important work. If it's successful at any level, it will prevent billions and billions and billions of euros that are used for healthcare for these diseases. The symptomatic stage is more difficult, and I will not speak about one of our other specialties of our teams at Harvard, which is to restore cell circuitry using neurons from stem cells. So, what about Parkinson's disease? Well, to get a little bit more complicated, but stay with me, Parkinson's disease, like many diseases, have a sporadic form. In fact, your risk, if you're a population, is 1. But many of you fluctuate about 1 to 2 to 3 to 4, 5 times the risk of your neighbors. Some are much more unfortunate They have a genetic predisposition, which make the risk, and we cannot calculate a number here because of penetrance. Some people develop it late in their life, and if they die earlier than that age, they don't develop the disease, but about 40 to 50-fold risk of an average person. Understanding these genetics now in humans allow us to basically make stem cells, iPS cells, from these patients and then study them in groups. Why is this so exciting to us? Because we can now define the mechanism of disease in a much more realistic and precise way. And we also hope, and our plan is, to understand how all these patients then, which is 90% or 80% of the population in Alzheimer's, Parkinson's, ALS, how they match onto these new subgroups. And also by drug therapy and screening, get much better medicines, much more precise medicines, and much more effective medicines. And to take that a step further, many drugs on the market, or even in the laboratories currently, are actually very, very good, but they fail in clinical trials. Why? Because the current reasoning and the current strategy, perhaps because we have no other strategy, is that one drug fits all. Even if you had a drug that was already developed and you had spent the money developing it, if you try it in a population of patients, you may have some patients that have no efficacy, they're non-responders. They don't even respond to the drug because they may lack the receptor for the drug. They have no toxicity either. Others are extremely sensitive and develop incredibly severe symptoms that may force the drug off the market immediately. Others may be good responders with low toxicity or acceptable toxicity. If we could define this curve, this Gaussian distribution, earlier in the process, we would be much more clever as drug discoverers. How do we do that? What do scientists do in the laboratories with these cells? Well, I'm going to give you a few examples. Again, let's recap. We have the adult cell, we have a reprogrammed cell, and we can make all the cells in the body. We have to do in the laboratory something that's akin to a disease simulation process. We will model the disease in a dish. Clearly, it's not an animal. In fact, that may be a disadvantage or an advantage. But nevertheless, we can do nothing to the cell. We can just study its aging process. We can use genetic forms of the disease. We can manipulate the cells. We can stimulate them. We can combine any of these manipulations in the laboratory. Scientists are very innovative and creative. The readout, meaning what we get as a result in the lab, can be defined as cell, organelle, and molecular phenotypes. And I will describe that in a minute. And these results guide us in our investigations. So what are the steps? First, here I'm going to show you patients' cells. In fact, I was leading a team from the National Institute of Health in the United States, which was charged in a much smaller way than IMI to generate cells from as many genetic forms of the disease as we could find worldwide. And this was the first initiative of this kind in the United States many years ago. And so what we discovered in a collaborative way, which was published in 2012, was that we could make those dopaminergic neurons that are so vulnerable in your brain in the dish. And that took us several years to learn how to simulate that process. You see the most vulnerable cell is actually here in blue because it has these markers. We can make the midbrain in a dish, we can sort the cells, and these are electrically active, and we can study them now in the way I described. So this is a representation of a cell in the body, almost any cell. It has a nucleus, it has all sorts of membranes that produce the proteins, it has mitochondria. Mitochondria are those little organelles in your cell that produce energy. They make the molecules that are used as fuel in your cells. And this particular cell also has a neuron, an axon, because that's what you have, and they make synapses in your brain. It turns out that in our first attempts, which we started in 2009, just after the discovery by Yamanaka, we could see all sorts of changes in these cells when we made them from human iPS cells into neurons. And they illustrated, and I just put in gray here not to overwhelm either myself or you, But instead today I will briefly speak about this aspect of the disease. We knew previously that if we made neurons exposed to something called mitochondrial toxins, in fact these are pesticides, forms of pesticides that in high concentration can get into a cell, they will selectively or with relative toxicity kill your dopamine neurons. Consequently, in rural areas with high exposure to pesticides, there's the 3 to 4-fold higher risk of Parkinson's disease. And aging also happens very rapidly in dopamine neurons and will affect these organelles. But for the first time ever, we could study this in a dish. And so we studied these specific responses and looked at what happened. I will show you, or try to explain to you, what we recently published in a collaboration. What you see on this left axis here is the number of damaged mitochondrial DNA molecules. You happen to have in your cell 2 sets of DNA, one in the nucleus, which is the shared combination of your mother and father's DNA on the chromosome, but also in the mitochondria you have a circular DNA, a little DNA circle that is carried from mother to child. That's actually how you trace lineage. So if you want to find out if you're related to somebody, what you do is actually study the mitochondrial DNA. You can't fake that one. Now what happens in your mitochondria, and you have hundreds of them in your cell, is that each time the mitochondria gets damaged, it will try to divide. Or if it's not functional, it may try to divide. So you have a population again, a Gaussian curve of functional and dysfunctional mitochondria. What we found when we developed these dopamine neurons in a dish from patients with a genetic risk for Parkinson's disease is that they had lesions, or what should I say, damage in the production of the mitochondrial DNA and RNA. For some reason, even at birth of these cells, they had problems making this mitochondrial DNA. And this was astonishing to us, but my collaborator, Timothy Greenemeyer in Pittsburgh, found that this was also true in patients' brains. So this process of mitochondrial trouble was apparent in the dish from these differentiated cells and also in the patient's brain. We could actually calculate the number of lesions or damaged mitochondrial DNA and production of RNA in this way. So what do you use these cells for then? Well, the scientists who are an aggressive bunch, I can tell you, for those who are not scientists, we try to attack the other people's work to see if it's true or not. And the way we do that is in scientific discourse sometimes, particularly well executed in conferences. But in the laboratory we do something. And what we do with stem cells is actually to create a correction of the mutation. So you basically fix the problem in the DNA by gene editing, and we did that here. And when we gene edited our cells, the problem disappeared. So in terms of a molecular diagnostic, we had evidence that this problem occurred in the patient's cells and we could correct it by editing. So the other aspect of this that I just wanted to illustrate quickly is that we can see in some ways what's wrong with the cell in the dish. And these were the dopaminergic cells from patients, actually a sibling, and in fact a set of related family members, in which one had a PINK1 mutation, that's one that creates risk for Parkinson's disease, young onset. And we predicted that those cells would generate a problem. And we challenged the cells in a dish with various oxidative stressors, also known in the public as free radical change. And to our surprise, and this is very typical in science, our first experiment worked, and then a lot of other things did not work. But I remember this very clearly when we took the cells, the neurons, and ran them through a column and studied their free radical formation after we had depolarized them. In the healthy subject, sibling control, we had a little bit of a signal. With the PINK1 mutation, we had a rather massive increase in free radical with the same stimulation. Cells were not dead, but it illustrates what I think is so useful with these human neurons in a dish. We can understand how the cells adapt and what the cells and the patient's genes do to harm the cells over time. Here's another illustration of this, and there's a lot of graphs on this slide. But what we also needed to understand is how useful are these cells? Why don't we use skin cells? They're also human cells, or blood cells for that matter. We have lots of blood cells that we can obtain from hospitals. But you see, when you study a disease, you really do want to study the cell that's most effective in the disease. Developing a drug from a fibroblast from a neurological disease may be possible, but maybe not appropriate. And here you see what we did. So we ran the same test on nerve cells from the patients, or from their own skin cells, fibroblasts. And as you can see, in these columns here, the nerve cells are much more sensitive. And you can also see that when we depolarize these cells, the pink one that I just showed you shows more cell death than the sibling controls. And so this is various ways in which we can understand these processes. In fact now, believe me, we're testing all these cells to try to find out biomarkers for a number of these diseases. So again, the 101 of mitochondrial biology and others is that we can stress the cells in very specific ways, but also understand how all these disease processes may lead to a common path. In a way, all roads lead to Brussels or Rome. In other words, a disease can start in any way in the cell, but in a specific disease, you usually end up with the same problem. And we can then distinguish the genes to a signature of what they do. And just as an illustration of this, we published not long ago that all these red blobs here are mitochondria in the cell. But just to illustrate to you that when we depolarize the cells and give an antioxidant, we can prevent the process that leads to the disease to some extent. It's obviously just a model, but it illustrates to you the idea that if you have a problem in the cell that's due to disease, you can find drugs to prevent it or even reverse it. Finally, I'd like to bring up another aspect that's not usually discussed, but certainly in the literature and something we do a fair amount of, is to use the cells in a transplantation bioassay. You could imagine that if you had diabetes, or you had a cell that would restore function in diabetes, you would transplant it first and restore function. And we do that in what we call a bioassay. Because cells in a dish are difficult to maintain beyond about 6 to 9 months. But if you transplant these cells to an animal, you can study them. And just to show you something relevant to Parkinson's, what we did here is we transplanted cells we have made into neurons from what we call triplication alpha-synuclein. Alpha-synuclein is the equivalent of amyloid in Alzheimer's disease. It generates protein aggregates. And the more you have of this protein, the higher the risk. And indeed, when we transplanted the patient human cells to a rat, here are the healthy subject, they grow axons. But we noticed something very interesting that was unknown in the disease mechanism, which is that this triplication gene delays the development of the neuron. So they develop less axons. These are the things growing out here. So this is things that we have now learned by studying human cells, not animal cells. And indeed, to illustrate this further, drug industry is now realizing that if we transplant these cells, what you're studying here is actually human neurons in a rat or a mouse, and the green here is the alpha-synuclein I just mentioned you. If you, and many drug companies are now doing this worldwide, try to reduce the amount of alpha-synuclein aggregation, you can actually give the drug to an animal and at least see if your drug works on a human cell. And that's something that we believe can be developed further, but obviously it's more of a future perspective. So, in so many words, I can summarize to you that the strategies for making disease in a dish, or model human disease in a dish, is now possible using iPS cells. And also, we will be able to, for many diseases, study predisposing factors in perhaps a more realistic way than previously. And I gave you an example of Parkinson's disease, which also applies for Alzheimer's disease. In fact, many of our diseases that affect aging, in which a predisposing phase should be looked at and treated early, and I believe iPS cells are very useful for that. And finally, like all things, there are pros and cons, advantages and disadvantages. And I'd like to, in order to also create a entrance to the panel discussion that we will have later and also to the next speakers to summarize what I've spoken about. Human-induced stem cells, iPS cells, from the same patient can show biological variability. This is great. All humans are different, and we can now understand more of a population approach to drug discovery. It's also somewhat of a problem because at some stage we need some consistency in our work, and this is a new challenge in terms of variability. Drug discovery and toxicity. Early risk prediction for new drugs reduces attrition and rates in cost. It's a great opportunity. We'll have to make that materialize, and we'll hear from Ruth McKernan later today about how realistic that is. But it certainly is something that I believe will happen once we have developed the tools. Cell autonomous versus non, that's a fancy word for saying what's happening inside the cell versus what's happening to the cell influenced by its neighboring cell. And certainly, iPS cells seems to be most suitable for doing genetic disease and understanding what happens to the cell in a dish. It may be not so clear in terms of what happens in a context or environment of other cells. But on the other hand, by transplanting the cells like I showed you, we may be able to get some of that information. And also mixing cells in a dish can be done creatively, even in three-dimensional structure, which I believe will happen in the future. Immaturity of differentiated cell. Well, it's great to have a human cell in a dish, but also it's pretty young. It's just a newborn cell. And with the nerve cells I study, they're basically completely fresh, and they've just done their final cell division. On the other hand, what surprised all of us in the field is that we could pick up disease so early. Or is it disease? Is it just what the cells do when they have different forms of mutations? They adapt. So by studying the adaptation, if you follow, in a dish, we may learn much more about biology and medicine than we have ever known before. Effects of genetic and ethnic backgrounds. Many diseases manifest what we call incomplete penetrance, and patients harboring the same mutations have different phenotypes. We have many genes in the body, and many ways in which those genes will be adapted to by your own machinery and cell and genes. So this is quite a challenge, but I believe this type of work is more realistic in the future. As I showed you, there are non-responders and high responders, and understanding those will give you an advantage. More knowledge is always better than less. Novel biological insight. A key challenge, of course, is to learn more from these cells than we can from other models. And I believe that's the case. In fact, we may hear today, there are certain diseases where we actually could not figure out a mechanism. And only when iPS cells came around were we able to figure out, for example, with rare forms of isoforms of molecular RNA editing, this is the way a cell makes proteins, we could only find that out by studying the human cell. So I believe that's a great opportunity as well. So with that, I'd like to show you the place where I work at Harvard, and there are many people that are involved in this and produce a number of fantastic experiments. We have also a number of collaborations throughout the United States and Europe. With that, I want to thank you for listening. Thank you.